So far, we've discussed computational workloads where the entire set of work to be done is known in advance, such as a set of frames to render or a set of simulations to be run. In this lecture, we're going to look at a more dynamic form of distributed computing, where you can execute multiple tasks, consider their outputs, and then generate new tasks based on the output of the old. This is known more generally as concurrent programming, and it has all sorts of applications in bioinformatics, molecular dynamics, and other fields. We're going to look at a variety of scientific applications that are created using the WorkQ concurrent programming framework that was developed by my lab at the University of Notre Dame. The first example that we're going to look at is Replica Exchange, which is a very common algorithm used in molecular dynamics. Now, the idea of Replica Exchange is this. Suppose I want to simulate the behavior of a molecule over a long period of time. As you may know, a simulation can run for a very long time to produce a very small amount of simulated time. I might run for hours in order to get femtoseconds of simulated time. So if I want to look at the behavior of a molecule, especially its behavior in unusual situations, I'm going to have to simulate for a very long time before I get useful results. Well, Replica Exchange is a solution to this problem. The idea of Replica Exchange is to run many simulations simultaneously at different temperatures. And the simulations that run at higher temperatures have more kinetic energy and thus sort of excite themselves into more unusual states. And so what we do in Replica Exchange is we run multiple simulations simultaneously, and we occasionally swap the states of simulations that are running at different temperatures. And the idea is to use the high temperatures to generate unusual states, and then bring them back to low temperatures to see how those molecules behave at those low temperatures. Now, in the past, Replica Exchange has been done in programming frameworks like MPI, where you have to have exactly n nodes running all at the same time. And as we discussed earlier, if you need exactly n nodes running at all the same time, you're likely to be disappointed. So what we did here is to express Replica Exchange using the WorkQ concurrent programming framework, such that we send out simulation tasks at multiple temperatures that run for short amounts of time, we bring them back, and then we submit more tasks. But because we're using a concurrent programming framework that can have variable concurrency, we don't have to worry about having exactly the right number of machines. And so we have a performance result here that shows as the number of replicas go up, the running time of our code is actually about the same. So we can get bigger and bigger and bigger, running more and more replicas without having a significant increase in runtime. And if we compare that to the old MPI way of doing things, it was just slightly slower than running it with MPI, but we're able to run it to a much larger system configuration. Here's another example, Genome Assembly. Genome Assembly is a critical problem in bioinformatics. As we learned in the first lecture, Genome Assembly is the problem of taking many different puzzle pieces of a genome that is acquired in the lab and figuring out how all those pieces fit together to create one long DNA string. Now, there are a bunch of existing tools out there to do it. A very well-known tool is called the Celera assembler. Now, in our case, this assembler was simply not running fast enough because its initial stages took too long to find all the cases where two common strings would overlap because they had a common sequence of letters in them. So what we did using the WorkQ framework was to accelerate the initial portions of the Celera assembler by parallelizing this initial alignment and overlap stage. So what we did here is we took these initial parts here, we caused them to divide up the overlaps into a large number of small tasks and farm them out to a set of workers, so that we could produce the initial stages of this computation much faster, and then feed the final stage into the consensus stage of the existing Celera assembler to get a result. And then when we compared the performance of this on a growing number of CPUs, the combined system was much faster than the original Celera assembler, and we were able to scale it up to nearly 1,000 CPUs and achieve a speed up of 952 times. My final example is the accelerated weighted ensemble. This is another problem in molecular dynamics where we're trying to understand the complex behavior of a protein in simulation. Now, proteins are kind of interesting because they're very complex molecules with a large number of atoms, and they can fold into a variety of different states of varying probability. We can call each one of these a configuration. Some configurations are more common than others. Now, for a given protein, we may or may not know the configurations, but what we really want to get out is the probability of that protein folding from one state to another. What we did with AWE is we designed a concurrent program to run a very large number of simulations simultaneously for a short amount of time, then to bring back the results and see if any of those simulations moved to a different state. After all those completed, we resample the simulations, send them out again, run for a short amount of time and see if they change state, and do this over and over and over again And what we begin to accumulate over time is a network of all the ways in which that protein can fold. So in this network here, each node in the network is a different configuration. Each edge represents a different transformation, and it's also weighted by the probability of that transformation. So as you've seen, a concurrent programming model like work queue can be used to create a wide variety of sophisticated and scalable programs. And in the next segment, we'll talk about the architecture of a work queue application in detail.